Good morning, and welcome to UCLA Department of Psychiatry Grand Rounds. We have a little bit of a different beginning. I am so proud and so pleased to announce the winner of the Laurie Altshuler Memorial Award. Laurie was the best of us. She was a multi-talented person, a world-class researcher, a gifted and warm clinician, a great mentor, and a teacher who nurtured young researchers and residents alike. And she was such a wonderful friend and colleague to me and to so many others in our department and nationally. Her research had both great scientific merit, but also addressed some of the most important clinical questions in mood disorders. Her research illuminated clinical practice as much as it did science. It is fitting that this award, named after Laurie, is given to a young psychiatrist whose early career shows the promise of contributing to our field in ways similar to that of Laurie Altshuler. This year's award is given to Dr. Laura Obler, who you see here, one of our fourth-year residents. Her nominating letter describes Laura as being among the top 1% of our trainees. She is a resident who provides exemplary clinical care, mentors junior residents, provides them with information, wisdom, and support. She works seamlessly with everybody on our clinical team, peers, colleagues, attending, social workers. Her commitment to patient care is incredible. As an example, she started a support group for postpartum women while she was doing her work in the Women's Life Center. And she has already co-authored a book chapter and has been planning further academic work for after graduation. I can say that Laura is also a resident in the Mood Clinic, which I direct, and where Laurie Altshuler was an attending. And I agree with all of these accolades about Laura. She takes care of very difficult patients with grace and beauty. It is a pleasure and a gift to work with her. I think Laurie Altshuler would have been thrilled to know Laura Obler. I am so pleased, Laura, that you're receiving this award.
Thank you, Dr. Gitlin. Thank you. That was very nice.
And now, Dr. Luchter.
Good morning. It's my pleasure today to introduce today's Grand Round speaker, Professor Marco Iacoboni. Marco is a professor of psychiatry and biobehavioral sciences and director of the TMS lab at the Amundsen Lovelace Brain Mapping Center. Marco trained as a neurologist and neuroscientist and came to the United States from Rome, Italy, where he joined the faculty of our department and the Amundsen Lovelace Center in 1999. There is a great deal of work that is going on in the Semel Institute and in the department, looking at brain imaging for the purposes of diagnosis of mental disorder, understanding the underpinning of disorders of mind and brain. And in that broad context of work, I think Marco's work is groundbreaking and is some of the most creative work that goes on at UCLA. He investigates the neural basis of sensory motor integration, imitation, and social cognition in humans, integrating fMRI studies and TMS studies. Marco has done groundbreaking work looking at the human mirror neuron system and its role in social behavior and social cognition and its disorders. His work has been recognized as being extremely important and well-funded by the NIH, the National Science Foundation as well, and of course has been covered in many of our most coveted scientific publications like the New York Times and the Los Angeles Times. He's an extraordinarily well-published researcher, really in top-notch journals. And in addition to that, he's also written a popular book, which I can commend to you, entitled Mirroring People, the New Science of Empathy and How We Connect with Others. I've had the pleasure of starting to collaborate with Marco, looking at studies of how we can use brain imaging and TMS to understand empathy and potentially to develop treatments for illness. And I'm pleased that he's here today to discuss his cutting edge work with us. So with that, let me turn things over to Marco.
Thank you, Andy, for your kind words, and thank you for the invitation. I'm going to share my screen. And I hope, Andy, can you see it?
Yes.
Good. So I assume the other people can see it too. So again, it's a pleasure to discuss with you some of my ideas about neurosystems for empathy and how we can study them to better understand how it works, but also to help mental health patients. This is the outline of what I want to discuss with you today. I want to say first why we care about empathy and social cognition. That seems, almost, of course you care, it's so important. But indeed, I want to make an argument, it's really important, even especially for mental health. And there are data that suggest that it's empathy deficits and social cognition deficits are really good predictors of major outcome measures. Then I want to talk about forms of empathy and something called dual process models that I'll explain what it is, but basically it's a theoretical framework that kind of explains some of the behaviors that humans have, especially when it comes to social cognition. Then I would like to talk about my early work in mirror neurons and how I got into studying empathy and how I got to study mirror neurons to begin with, which is many times in life when things are important, it's just sort of serendipitous. The bulk of the talk is about what we've been doing in the last 5 or 6 years or so, in which we were studying how systems for affective and cognitive empathy are actually very well integrated, in spite of the general idea that they tend not to be. And I want to finish with the latest works in which we're using machine learning to really predict dispositional empathy just by looking at connectivity in the brain. Okay, let me start with why we care about empathy. We have this thing that we call social embeddedness in my group. And basically the idea is that you need to be embedded in your social milieu, your social context. Because if you don't do that, then human flourishing, which is the term that comes from really all the way back to Greek philosophers some 3,000 years ago, it's hard to achieve. Human flourishing really requires that you really fully and fluently and smoothly embedded in your social milieu. And social cognition, which is that set of cognitive skills that are useful to be a competent social agent, it's really important for achieving this social embeddedness. And I think that pretty much everybody who studies social cognition, which is a complex thing, would agree that empathy is one of its core functions. That's why this is so important to study empathy. And recently, we became more and more aware of how important is social cognition, because there's plenty of data now in the literature that show that social cognition deficits become, and I'm using the words of this review article that came out some 3 years ago, a core phenotype of many clinical conditions. And this article really lists psychiatric, neurological, environmental conditions in which social cognition deficits are really important and prominent. Just to give you some examples, social cognition deficits can predict symptoms of mental illness, disability severity, and even increased use of mental health resources like hospital admission, emergency visits. These are really big outcome measures. And they're also important from the standpoint of being a burden to our healthcare structure. So it's really important to try to understand how social cognition works. And it's not easy, admittedly. It's a very complex thing, and try to fix it. Because if you fix it, the belief is that indeed you're going to actually see improvement in the clinical status of many of these patients. And now let me talk about kinds of empathy. Scholars and scientists who study empathy may spend an entire meeting just to discuss the various forms of empathy. But I would say everybody agrees that there are 2 main categories or types of empathy, affective and cognitive. I don't know if you've ever read this book called Emotional Contagion. It came out almost more than 25 years ago, I think in 1984. And really shows the many examples in which humans can catch the emotions of other people, like we catch viruses. And that's really the kind of affective empathy that we tend to study, and it's easy to study in the lab, and I'll show you some examples of that. Then the other type is what's called the cognitive empathy, and this is the classical and very famous developmental test of the Sally and Ann test, in which you really have to understand. So the story is that you have this girl that is putting her doll somewhere in the room, then she leaves the room, and the other girl moves the doll somewhere else. Then the first girl comes back, and she's looking for a doll. You really have to understand what this girl knows and what she doesn't know, to really predict where she's going to look for a doll. So that's very, very cognitive. Emotions here are not really much in the picture. So this is just to give you two examples of these domains of affective and cognitive empathy. Now, when it comes to brain imaging and neural systems for empathy, there seems to be also some kind of nice separation between systems for affective empathy and systems for more cognitive empathy. This is a review article that Jamil Zaki and Kevin Oxner published almost 10 years ago in Nature Neuroscience. And this figure really shows basically a summary of many, many imaging studies that seem to suggest that indeed the areas in green are the ones that have to do with affective empathy. And we're talking about premotor cortex, especially its inferior part, the inferior part of the parietal lobule. These are really the classical mirror systems that we've been studying in the lab for about 20 years now. Also, the posterior superior temporal cortex, it seems to be the area in which these frontoparietal areas for mirroring get the visual information from. So this is the entry point, the visual entry point for mirroring systems. And then also the anterior cingulate cortex that seem to be active when we feel pain and when we see other people in pain. So these are all forms of systems that have to do with affective empathy, with the kind of empathy in which we do some sort of a neural resonance, in which whenever we experience something, we see some other people experiencing something, there is a co-activation of these systems in these two conditions. But then there are also these systems for more cognitive empathy, including the medial prefrontal cortex, the medial parietal cortex, the temporal parietal junction, and the temporal pole. So if you look at the imaging literature at the level of neural system, there seems to be also some nice separation between affective and cognitive empathy. Now, let me tell you something about this idea of dual process models. What are they and why they were invoked? Well, mostly they were invoked to explain some really odd behavior, such that people overtly reject being prejudiced, and that if they are performing a task in which they can show some implicit prejudice, then what's explicit is basically denied. I'm not a prejudiced person. Through this implicit task, you can actually show implicit bias toward other people. Why is that? Why do we have this kind of odd behavior and then sort of contrasting behaviors in these two situations? The idea is that there's some knowledge that is stored in this associative network, which is automatically activated, influences our thoughts and our behaviors, and it's fairly independent from more deliberative judgment. These are really words from David Amodio. He used to be a postdoc here many, many years ago, and he's a trending current science article came out a couple of years ago. And if you want to know more about it, you may want to read Thinking Fast and Slow, which is a lovely summary of Dan Kahneman's work that led him to get the Nobel Prize, in which really the idea of the fast thinking is this automatically activated association memories. And the slow one is the more deliberative judgment. So but why, if I'm saying that I'm not prejudiced, and I run an implicit test, I may show prejudice? The idea is that, again, the associative network, fast thinking, this one, it's controlled by the more deliberate processes, covering control areas. And yet, in some conditions, especially conditions that do not really require us to be deliberate, especially in very simple tasks like the IDLT, then the second process, the control process, basically fails to model the first one. And that's when our implicit biases come out. So that seems to suggest that control processes are good, and our implicit association memories are necessarily or seem to be bad. That's not exactly what led Paul Bloom, which is a very famous and super smart psychologist, to write his book, Against Empathy. They came out about 5 years ago. And when he says against empathy, he really means affective empathy. And indeed, if you look at the subtitle of the book, it's the case for rational compassion. He's basically making an argument that empathy is bad for you because, and again, he's thinking mostly affective empathy, mostly because you get swept away by emotions. And so you're not really making good decisions. Whereas if you have rational compassion, then you're going to make good decisions that can also be pro-social. Some beg to differ, that's including me, but I would say that I want to cite John Badge's work. And this is a paper that came out also in Planetary Science in 2018, entitled The Mythical Number Two. And when he says number two, he's really thinking about these deliberate processes, the slow processes, the slow thinking that Dan Kahneman's book refers to. And I'm quoting for the article, perhaps the most persistent fallacy in the perception of dual process theory is the idea that type 1 processes, intuitive, heuristic, are responsible for all bad thinking, and that type 2 processes, reflective, analytic, necessarily lead to correct responses. And I say, well, we often fail because type 2 thinking produces self-sabotaging rationalization. And I would say here, I would translate this mostly motivated reasoning. We have many examples these days in the public discourse of motivated reasoning that lead to very bad conclusions. So they keep going and say that basically effective self-regulators are those who rely on type 1 processing, which basically means effective empathy, fast thinking, to regulate their behavior, not type 2, which is the deliberate thinking. So here we have two contrasting situations, right? So on one end, you can, especially in the domain of prejudice and bias, it seems that deliberate processes tend to lead you to good conclusions, whereas the fast thinking may actually reveal your biases. But here there's also an argument for the opposite, that indeed self-regulators are those that rely more on the affective component rather than the cognitive one. Okay, now let me tell you how I got into this whole thing. I was in 1995 in Prague, of all places, and I was at a meeting showing some of my work on sensory motor integration. That's what I really wanted to study, how the brain puts together perception and action, which is something essential in our life. We do it all the time. And I ran into Rizzolatti. He was at the same meeting. This was a year before the famous Gallese paper came out in 1996. And he told me about these cells. And he was saying, you know, in the same cell, we see basically the perception action link in just the file of a single cell. I was a little astonished, also a bit in disbelief, but I started working with them. And we get funded, and we started doing brain imaging studies on this line of research here at UCLA. I was already at UCLA when I met Solati. And when I started doing my work on mirroring, I thought, okay, if we have mirror neurons in the human brain, what do we need them for? And it seems to me that imitation was really a fairly low-hanging fruit. Imitation, at that time when I published this paper in 1999, no one had ever done a study on using brain imaging on imitation. And the idea was that that's because imitation is such a complex thing. It's difficult to make it tractable in the lab. We were very successful in doing that, and we published many other studies on different components of both the neural systems and the functional mechanisms of imitation. In the same year in which I published this third early paper on imitation using brain imaging, John Badge and Tanya Shatran published this paper in the Social Psychology Journal, in which what they show is that when we interact with other people, we tend to spontaneously imitate each other. And the more we tend to do that, the more we tend to be empathic. I have to thank Celia Hayes, which is the famous British psychologist who studied imitation all her life, to send me this paper. She had read my science paper on imitation, and she sent me this one. And then I started thinking, okay, but how does it work in terms of neural systems? Of course, I was already well entrenched into studying neurosystems for mirroring that to me were important for imitation. But of course, empathy also requires emotional involvement. And then I thought, well, perhaps at neural systems level, and we basically started collecting data for this study about exactly 20 years ago in 2001. The idea is that, well, I see a nice smiling face. My mirror neurons stimulate internally that facial expression. My neurons that control my facial expressions activate mildly. I do not mimic the face, but simply internally there is the activation. But somehow that information needs to be transferred to my emotional brain systems. And I was looking at the anatomy and I looked at the insula. Indeed, it was a very good pathway to connect our mirror neuron systems with the limbic system. And so the idea is that you see someone else expressing emotions in their face, your mirror neurons stimulate that facial expression in your brain, and then information gets spread into your emotional brain system such that you actually feel the emotion developing. This was the main model I wanted to test in using brain imaging. We tested in this study in 2003. At that time, we only had, and the data were supporting the model, but at that time, we only had brain imaging data. And then after that, many other labs, including us, what they did was to use similar paradigms, but also correlate those with behavior or even real life behavior. This is a study that we had done that was led by the other director here in Los Angeles, and we were recruiting adolescent kids, and we were studying for many years, including, and we're doing, among other things, also our mirroring tasks in the scanner. The kids were activating exactly the same circuit I told you earlier, mirror neuron areas, the insula, all the way down to the amygdala. But now we have also these behavioral reports. This is an interpersonal comments. It's a report from the parents of the kids. And they show that indeed, if we correlate brain activity while the kids are imitating or observing this facial expression with their interpersonal competence score that is filled out by the parents, there is a very nice correlation such that the more they tend to activate this old circuitry when they see or imitate facial expression in the lab, in the scanner, the more they tend to be socially competent agents. So that really suggests that all this affective component of empathy has already some links in more deliberate sort of behavior, because you want to, of course, conceive that when it comes to this being a socially competent individual, you do require deliberate thinking and judgment. And the other thing is that I was thinking about is that, well, it's possibly true that many brain imaging studies seem to suggest some kind of a separation between systems for affective empathy and systems for more cognitive empathy. But on the end, we know that the brain systems are heavily interconnected. So could it be that indeed there is more crosstalk than we think there is when we just look at levels of activation? And this is what I want to talk about now, basically what we've done in the last few years, in which we're using both brain imaging and brain stimulation to try to test the idea that maybe it's not really a dual process, just one. It may look two, but indeed it may just be one. It's like when you look up in the sky and you see the sun moving on the sky from the morning to the afternoon. You may think that it's the sun rotating around the earth, but it's actually the opposite. So could it be that indeed appearances here are actually telling us something that is not necessarily entirely true? So we decided, let's do a series of studies in which what we do, we have participants, we put them in the scanner, we do our classical mirroring, affective, emotional empathy sort of tasks. They see facial emotional expression, they imitate them. They see someone experiencing pain. This is a screenshot of a video in which the needle is going to pierce the hand and they have some control condition. We measure brain activity in response to this sort of affective, emotional empathy kinds of tasks, pre-reflected. They don't have to make any judgment of any kind. Then outside the scanner, they play an economic game. The game is such that they play with 24 strangers. They don't know them. And actually the name and the face have been changed for anonymity purposes, but they know that there are real people in LA, and they get given for 24 trials, $10, and they're told you can keep all $10, or you can share some of this $10, or even all of it, whatever you like. It's called the dictator game, because you basically decide everything in this game with these people. And the only information that you have is that pretty much what's the gender and age, because even if the face is a different one, it corresponds to the gender and face and race of the individual that is out there. The name is being changed, but what's not being changed is the amount of money that these people make in a year. And they play with players that make very little money and players that make much more money. But the idea is that, and why we do that? We do that because we want to show that our participants are actually thinking. That is, if I'm playing with this dude and I want to share some of my money, of these $10 with him, and then I play with this other guy who makes twice as much, 6 times more the money that this guy makes, I'm likely to be more generous with the first one than with the second one, because the second one on the face of it doesn't really need my generosity. And that's exactly what our participants do. They really show that they are thinking about their decisions and their generosity. The question is, can we use brain imaging to predict the level of generosity in the game that our participants are showing in their decisions? And the answer is yes, we have two types of correlation: direct correlation between brain activity when you see, say, someone in pain and your tendency to be generous, to share money. And if they really are in classical affective areas, there's the activity in the amygdala. The more your amygdala gets active when you see someone in pain, the more you tend to be generous in the economic gain. But also inverse correlation between activity, brain activity, and the tendency to share and to be generous. And this really seems to be mapping well on cognitive control areas like dorsolateral prefrontal cortex and TPJ. We also get the medial prefrontal cortex. And here the idea is that there's an inverse correlation in the sense that lower is the activity in these areas when you see someone in pain, and the more you tend to be generous. The higher is the activity in this area when you see someone in pain, the less generous you are. Note also that these regions, they do not tend to be activated much by our simple mirroring task, which really suggests what these correlations are doing, are really tracking individual differences in levels of control. So we find a correlation, but as you know, correlation is not causation. My favorite example is that you can find in books of statistics, that you can get a bunch of cities in the United States, and you can count the number of crimes and the number of churches, and lo and behold, you find a nice correlation between the two. But what's the causal relationship? Well, the causal relationship has nothing to do with crimes and churches per se, but only with the level of population. In bigger cities that are more crowded, there will be more crimes and there will be more churches. In smaller cities that are less crowded, there will be less crimes and less churches. So one needs to be careful with correlation analysis. Luckily now we have this tool and others in this category of tools called non-invasive brain stimulation, in which we can target the system and sort of disrupt its activity and see whether or not it does change behavior. So we did a follow-up study. In this follow-up study, we were targeting the dorsolateral prefrontal cortex, which did show those inverse correlation between activity and generosity. That is, the more activity, the less generous our participants are. Dorsomedial prefrontal cortex showing the same inverse correlation. The more activity is, the less generous they tend to be. And then a control area. One of my favorite control areas, motion processing area, the MTV5 region. And this is a control region we need to have to really just control for generic, non-specific types of effects of brain stimulation. Here is the prediction. So they get stimulated with this thing that we call theta burst stimulation. It's a very powerful form of stimulation. It's the continuous form, so it's the inhibitory form. So we're basically inhibiting these three systems when we stimulate our participants, 3 different groups. And then after they get stimulated, they play the game. The effect of theta burst stimulation lasts for about an hour. So it's a very handy way of doing brain stimulation that perturb brain system and may change behavior that you can test over a fairly long period of time. So they play the game again And now the idea is that if the correlations we saw in the early study were causally related to the generosity of our participants, by knocking down these two regions, by reducing their influence in their behavior, we should make people more generous. Because remember, these areas are areas in which the more active, the more engaged they are, the less generous people are. If we knock them down, we should make people more generous. If there is a causal relation between the activity in these areas and their generosity, but not in the control area. That's what we show in this follow-up study, in which we actually see that these are the offers made by the participants that were stimulated in the dorsal medial prefrontal cortex, in the dorsolateral prefrontal cortex, and in our control region. And these are offers made to people that make quite a bit of money. As you can see, again, our participants are still thinking, even though they get that in the brain with brain stimulation, because they are much less generous with people that make a lot of money compared to people that make very little money. But crucial to our hypothesis, you can see that there is an increased generosity for people that make quite a bit of money in the dorsolateral prefrontal group, which suggests that what this region is doing is really contextual processing. I'm looking at someone's face. I know how much money he makes. And I tend to be a little stingy if he makes a lot of money. But if my DLPFC region is disrupted in its activity, then I become a little more generous. And the other one for participants that are generous toward people that make very little money, what you can see is that the strongest effect is really in the dorsal medial prefrontal group. And here the idea is that the dorsal medial prefrontal group provides some sort of a tonic inhibition that is not really processing contextual information. And there's more evidence for this also from brain imaging studies about that. Okay, so now we've shown that our correlations in the first study, thanks to brain stimulation, it wasn't just spurious correlation. There is a causal relationship between the brain system and the brain activity that we saw activated and predicting generosity in our game. And that's something I really want to try to emphasize. This is a nice way of combining brain imaging and brain stimulation. It doesn't have to be done online. It can be done offline. But it is that you can have this model using brain imaging that provides some correlation information. And then using brain stimulation, you can perturb the model. You can extract causal information about how you can disrupt local activity and then predict specific behavioral consequences. And taken together, this study really suggests that affective empathy and cognitive empathy are continuously interacting and may form an integrated processing stream, even in tasks in which you don't think you need them. Basically, the idea is that we actually find correlations with DLPFC, which is not really activated when I see someone in pain, particularly not much activated. And generosity in a game, it suggests that that brain activity in some way, even though that system is not really strongly engaged in the task of seeing someone in pain, it is still tracking some level of activity. And that's why the notion is that these two systems in the brain are continuously interacting. But can we test this hypothesis even more? You may know that one of the most original contributions of brain imaging is the discovery that we have these systems in the brain, even at rest, these functionally organized systems, and most of them really mimic task-related functional organization like visual, sensory, motor, auditory. And indeed, there are studies that really suggest that there is a nice link between resting brain activity and activity in the brain during tasks. Now, the question is, if the system for affective empathy and the system for more cognitive empathy that we have seen are actually interconnected and cross-talking to each other across tasks, are really strongly integrated, can we actually see that at rest? Can we measure brain activity at rest and predict self-reported empathy? That's what we did in the study that we published last year, in which we basically get a bunch of ROIs, resting state scan. We get a bunch of ROIs driven mostly by your own empirical work, but also by the vast literature that is now on affective empathy and cognitive empathy. And we use these ROIs, and then we track the level of connectivity at rest. And then we use machine learning to basically train a classifier, and tell the classifier on a part of the data, this is the level of, this is the pattern connectivity, and this is the self-reported empathy that these participants are reporting through a well-validated questionnaire called the interactivity index. And then now that you've seen these patterns, can you actually predict just by looking at connectivity, the level of self-reported empathy in other participants? And we do this with many iterations. And what we do find is indeed that the classifier, basically an algorithm, can predict self-reported empathy simply by looking at connectivity between functional connectivity between our affective empathy neural systems and our cognitive empathy neural systems. And when I got that, I thought, well, but how does it work? How is it that just by looking at these dude face and how much money he makes, and this crazy scientist gives me $10, and I can share some of these dollars with these strangers, and I know they're going to get the money in the mail, because that's a key aspect of this whole thing. If participants need to know that someone is going to have a real world effect of their own decisions. How is it why my response in the brain when I see someone in pain predicts my level of generosity? And the way I was thinking about it is that, well, maybe what happens here when we think about people, because even when you see a neutral face, you're still thinking about someone, right? You're thinking, well, this guy makes $25,000 in LA, not a lot of money for LA. When you think about people, then you're thinking, even if it's not, super deliberate, in some way it's shaped by your previous experiences and your memories of how you interacted with other people. And this goes back to the whole idea of the dual process model that I told you earlier. That in some way, some of these implicit memories are not necessarily bad, can actually shape a good decision making. And I had some evidence in my own work. I had this work that I did in collaboration with Iacoboni, in which we were studying these neurosurgical patients that they were undergoing surgery, and before they were undergoing surgery, we could test their responses to our classical mirroring test in someone observing or grasping something or making the action, or seeing a smiling face or making a smiling face in a control condition. They have electrode in the brain. We can measure brain activity. We were looking for mirror neurons in a human brain from the signal unit activity, spiking activity. And we were recording from the medial prefrontal, from frontal region, especially in the middle wall, and from medial temporal regions too. I was expecting perhaps to find some units to mirroring properties in the frontal lobe, but certainly I wasn't expecting to find anything in the medial temporal lobe, which is a system in the brain that's very important for memory. It's not really famous for or known for motor properties. One day, Roy Mukama, which is the postdoc that was the lead author of this study, who's now a professor in Tel Aviv University, comes to my office and shows me this cell that has a nice mirroring response for seeing or making a smiling face, for seeing or making a frowning face, but not for the control condition. And these are single unit activity in humans, which is one of the coolest data that you can possibly deal with. And he tells me, Marco, I found this cell in the entorhinal cortex. And I told him, Roy, are you drunk? How is it possible? Those regions are not famous for and known for motor properties. And initially I was really misbelieved, but then we've seen this over and over again. And here the idea is that what's going on, it's not only, your brain doesn't just mirror the actions or the intentions of others or the emotions of others. But what can happen is that these areas are also, these medial temporal areas, are areas that record your own memories. So when I'm grabbing a cup of coffee, my medial temporal cells are recording my own actions. And when I see you doing the same thing, I also reactivate the memory trace of myself doing that action. So that really mirroring, it's a much deeper and richer way of connecting between people. And we have this beautiful study that came out very recently from Nadia Sutana's lab. She's such a star. She's always been a star. She was a grad student, but now she's doing this amazing experiment in which she has this patient with implanted devices, and she can record activity in the brain while they're walking around and seeing things. And they report indeed similar activity and EEG activity and electrical activity when the patient is making certain kinds of movements in an environment, and especially getting close to the wall of the environment, and also when seeing someone else doing it. Again, here mirroring deeply embedded in memory areas. And that really suggests these are devices are generally recording for medial temporal lobe. And so the idea is that again, these memory systems may be a modulator of the integration between the affective and the cognitive empathic system that we discussed earlier on. Okay, where do we go from here? One thing we want to do is to use better predictor. Again, I just told you one example would be to add in our machine learning work to add more systems, including medial temporal lobe areas, more refined predictors. The ROIs that we use in our study last year, some of them may not be relevant, and some others that are relevant, we miss them. That's because the literature isn't necessary. We don't know everything about systems for cognitive and emotional empathy. Then using multiple imaging features, especially diffusion MRI, because, and we're going to make an argument about using those for treatment outcome, especially PMS effects, they spread through wide body tracts. And so having information about diffusion data is very important. And possibly also using more scalable features like EEG. Because if we have good predictors of empathy through more scalable features, that's also useful since imaging is very expensive. We want to expand on task. We already done some work on moral dilemmas. I haven't told you about that because I can't pack too much stuff in 1 hour of a conversation. We also want to use naturalistic fMRI. Naturalistic fMRI is something in which we basically see a continuous input of like a movie, and you track the inter-subject correlation between them. Recently, they really seem to suggest that it also tracks group identity, group membership, which is an important aspect of empathy. We tend to empathize a lot with people of our social group. We empathize much less with people of a different social group. And that's something it's important to understand, to better understand how to intervene. What we want to use brain stimulation to understand better the computational architectures. And I'm not going to unpack this will require some other talk entirely, but basically the idea is that you may conceive different computational architectures that may respond differently to different kinds of stimulations, whether it's excitatory or inhibitory. And this whole exercise, it's really to try to understand whether we can use this information for prediction of treatment outcome. I mean, TMS is it's been now heavily used in depression, but still there's the sense that it's not used as better, as good as it could be. And being able to predict treatment outcome, especially using brain imaging or EEG parameters, may be very, very important. Okay, let me conclude. So when we think about people, that thinking is really shaped and grabbing onto the perceptual and motor experiences, the memories that we had when we interacted with them. And this work really does, the work I've made in the last 5 years, seems to suggest that affective and cognitive empathy are continuously interacting, even in tasks that seem to require only one of them, like seeing someone in pain or thinking about being generous with someone with more or less money. Indeed, we can actually use resting state connectivity and looking at those patterns between affective and cognitive empathy and predict empathic predisposition. And a longer term goal is really how to understand how it works in great detail, how neuropathology and psychopathology change it, and especially possibly to come out with predictors of treatment outcome for brain stimulation treatments in mental health. And my concluding slide is to acknowledge all the people that and the funding that are doing this work. Thank you so much.
Thank you very much, Marco, for a terrific talk and really very, very interesting research. We've got a couple of questions coming in here, and I did want to encourage everybody to go ahead and submit any questions that you might have that we can address during the Q&A period here. Marco, I wanted to start off with one particular question. Looking at some of the changes that you're able to induce with TMS, like, as you said, creating an altruist through brain stimulation. I wonder if you could comment on, number one, the durability of these kinds of effects that you're seeing. The potential to try to make some of these changes that we're looking at, changes in disposition, changes in empathy, more enduring, and whether there is a path towards better therapeutics through those kinds of modulating those kinds of mechanisms.
Yeah, so that's an important and great question. Of course, one example, a very simple first order answer would be the more you do, the more you stimulate, the longer is the effect that you're going to get. That's why when you do when you do treatment of depression in the multiple sessions, it's unlikely that you can actually have a long-lasting effect if you just do one shot. So the dose is certainly important. I would say that what's also important is possibly to come up with more creative ways of doing brain stimulation, for instance, concurrently with some tasks, so that we can induce a mental state. Because when we stimulate, when the patient comes to the clinic to treat depression, for instance, there is an ongoing activity in the brain that we're going to stimulate. Potentially, and there is data that suggests that if you actually can put the brain in a given state at the time which you stimulate, you can get stronger responses. So that could be another way in which you can actually have more long-lasting effects. Seems to me those are the main important ones, basically the idea of dose and the idea of what the brain state in which you stimulate. There are other things to consider. One is all this notion of personalized treatments. So people that want to actually try to achieve very precise form of stimulation. I'm not entirely convinced that that's going to give you such a big payoff. But certainly the other notion that we try to advocate with this work is the notion of, can we have predictors of treatment outcome before we can even start the treatment? So that we can use brain imaging to, and having, of course, more information on this, but having brain imaging information to kind of tailor the amount of dose that you need to give to a participant to a study in order to have a much longer lasting effect.
Right. Okay, very good. Thank you. We were talking about brain imaging a moment ago, and we have one question here about some of the different imaging techniques. The question is, isn't PET superior to fMRI to reveal brain dynamics of both cognitive and emotional perception action cycles? And I suppose it's sort of to expand that a little bit, I wonder if you might comment on some of the other techniques that can be used to study these processes, including not just PET or fMRI, but EEG and potentially other methods.
Yeah, I think that's also a great question. I think that we have all this arsenal of tools and we want to use them all. In fact, we've been waiting for a long time for having a PET MRI machine and system here in brain mapping, and the whole process has been stuck in Sacramento for reasons I don't understand. I would say PET gives you complementary information. Joachim, it seems to me that with fMRI you get a much better temporal resolution. With PET you get very important information that has to do with neurotransmitters. You can also use it to sort of be more precise when it comes to especially distant effects. One nice way of doing PET in combination with brain imaging, with brain stimulation is, for instance, to use stimulate the cortex and see effects of ligands that you can use in PET studies down in subcortical structure. That's something that with fMRI you cannot do at all. So that's certainly an important component of looking at the interaction between systems, especially when it comes to brain stimulation. But it's not the only one. And again, the beauty of fMRI is that it gives you a lot of temporal information that you can use for this, especially for this functional connectivity analysis. And the other thing is that with MRI in general, you can get information on white matter tracks that it's not easy to get with other kinds of imaging techniques. So that's what's important, because let's not forget that TMS was really born as a motor physiology tool, so that I stimulate my left motor cortex and I get a twitch in my right hand, and that's because my M1 neurons go all the way down, and that affects to the cortical spinal tract, the anterior horn of the spinal cord, all the way to my muscle. But really, the effects of TMS do spread through wide matter flexion, so that's another important one. But yeah, EEG. Of course, EEG would be great to have EEG predictors of treatment outcome. It's much more user-friendly. It's much less expensive. So it'd be great to do that. And that's another thing that we certainly want to use in this work to initially to compare with brain imaging, and possibly if we see that we can get away without using brain imaging, that would be even better, because then it becomes way more scalable.
Right. Okay, great. Along the lines of functional connectivity, can you comment on the relative importance of functional versus structural connectivity and the heterogeneity in structural connectivity, and how that may contribute to, how that may influence some of the findings here?
Well, yeah, structural connectivity has been, at the center of many controversies in the imaging world, because there's been plenty of these things. But certainly, it's one of these parameters. It's the only thing. Diffusion is the only thing we have basically to quantify one matter. There may be issues with some of the parameters we use, but it's certainly important. Initially, there were data suggesting there was a nice link and coupling between functional and structural correlations. May not be necessarily the case, and we may get certainly more information using both, and especially with this multivariate statistics approach that we're using now, which are very, very powerful, having more features that can be used, especially for prediction, is especially important. I would say now we tend to do always multimodal imaging. We like to do resting state, task-based, if we code for it, and diffusion pretty much every time we have people in the scanner. Of course, diffusion will add more time, but it can be a very important type of information that you want to add.
Okay, great. Changing topics here a little bit. We have a more general question. I think you've touched on some of this, but the question is, what does your research tell us about diagnosis and prognosis in addition to treatment?
Yeah, well, that's at this time what we tend to do is to get information from our participants, so then we can actually use the behavioral information and correlate that with brain imaging. But I can see a time in which we're going to have such a plethora of brain imaging data that in principle we could use brain imaging data even to make diagnosis or prognosis. And I'm really especially thinking about mental health, because the reports you get from mental health patients need not be necessarily accurate or something you can necessarily trust. So it'd be great to have, say that we are going to study the effects of an intervention that really tries to ameliorate empathy in the order to then ameliorate social cognition in a group of patients. And say that the reports of this patient may not be entirely trustworthy, especially early on in the intervention. Then it'd be great if we could use brain imaging to see whether or not we are actually really sort of steering the brain dynamics, the network interactions toward what we want to send them, so that it seems to be promising when it comes to really having a clinical outcome. So in that regard, certainly, this kind of research may be very important for both diagnosis and prognosis beyond treatment.
Okay, terrific, thank you. We have another question here, commenting on the concept of social embeddedness that you spoke about in your talk. And one of our participants asked, why do you assume that social embeddedness is essential if many very successful people did not have it and did not appear to suffer from a lack of social embeddedness?
Well, yeah, I mean, certainly it's not a one-to-one mapping, but I would say statistically speaking, most people that tend to be, again, happy and flourishing are people that tend to be very well entrenched in their own social media. There are exceptions, and these people may be exceptionally talented individuals. They may not need to have social support and still be fairly successful. But first of all, I'm still not convinced that even in these particular cases, success doesn't necessarily mean flourishing. And that's why I'm using the word flourishing. Because if you go back to the Greek philosopher, flourishing has to do with more than security, way more than that. But also, again, we are making more like a probabilistic argument, in which exceptions are, of course, accepted. There will be exceptions for sure. I think that probabilistically, social embeddedness kind of predicts flourishing.
Right. Okay, very good. Well, that's about all the time that we have for today. And I want to thank you once again, Marco, for a very stimulating talk and for sharing your work with us. And many thanks to all of you for attending today's Grand Rounds. Have a great rest of the day.
Bye.